Welcome back to the course Fundamentals of General Immunology, and we are so this is our lecture number 12, intracellular antigen routing and processing pathways. So in the last class what we understood is how these antigens are processed, and we also discussed about two molecules, MHC1 and MHC2. What are the proteins which are involved in making these two molecules. So MHC1 we discussed in detail, MHC2 also we discussed, and in addition to 1 and 2 we also looked into MHC3, which is a gene which is a protein coded by a gene that is responsible for production of multiple cytokines including TNF and complements. So let us move on and try to understand further the pathways in details. So let us try to understand the two main pathways which we are discussing. The first one is endogenous pathway and exogenous pathway. Just to recapitulate the discussion again, that the endogenous pathway is the one which is coming from the produced antigen inside the cell. So the cellular proteins are produced inside and they are processed through this molecule called MHC1. So we will try to understand this pathway first, and then we will move to the exogenous pathway. So we also discussed that these MHC1 molecules are present in each and every nucleated cell. These are highly polymorphic in nature, and we also looked into their location in chromosome number 6, where there could be possible 3 different types of MHC1 molecules, which also called as HLA types, that is giving a variation in these molecules, and accordingly the proteins are the antigens are processed through this MHC1 molecule. So let us move on and try to discuss this pathway in detail. This is very important pathway to express or to show the viral specific antigen to the human immune system or the immune system or the surveillance system of the human body. So let us try to briefly understand one important point, that when there is a binding, so suppose this is an MHC1 molecule. So what we could understand that this is a beta 2 microglobulin. We can simply write beta 2M, and these are alpha chain, right. So this is alpha 1, alpha 2 and alpha 3, and these are the peptide or the antigen coded molecule. So we have a similar kind of receptor expressed by the T cell as well. So if there is a recognition by a proper shape conformation, then we can see that there is an activation pathway, and if there is no recognition because the structures are quite different and this MHC may not able to bind properly to the antigens expressed antigen or the antigen of concern. So if there is no binding, we do not see any activation, there is no recognition, and therefore there is no activation of the T cell. This is just a cartoon to show that the antigen variation exists in the domain of MHC. Antigen binding variation exists in the MHC1 domain. Here also you can see that the compatibility is not perfect because of simply the types of antigen which are there. So that stands for two things here, that the size of the antigen or the conformation of antigen is very important to bind with the MHC1 molecule. Second, the type of MHC1 molecule is very important. So suppose in this case we have two similar kind of antigen, but this HLA types are different, and therefore we can see that MHCA and MHCB are different, and therefore the binding of these two antigen may not be recognized by the MHC molecules. So the type of MHC is important, and the second is conformation of antigen is also important to start the recognition of the T cell or the TCR binding. TCR stands for T cell receptor, which is here. This is also alpha and beta chain, which we will discuss a little later, and we could look into the biology of TCR in context of MHC1 expression in this endogenous peptide structures. So let's move on and try to see this pathway in clarity. We will go in detail about that, but let's try to understand this whole pathway in context of its processing of the antigen into the cytoplasm. So what's happening here is, suppose we have a cytosolic protein, and these cytosolic proteins are having these epitopic markers which are present or exposed to the surface. These are taken by the cell through multiple pathways, and these antigens are then processed through this proteasome. Now, proteasome is a structure in the cytoplasm that is known to degrade the larger fragment of protein into smaller fragments. So, proteasome is a cellular organelle. Remember, this is present in the cytoplasm, and the function of proteasome is to break down the larger protein into smaller protein, and this can help in assisting the salvage pathway. Now, salvage pathway is something which we will study later on, where the degraded product of the proteins or say other biomolecules can be recycled to produce a new one, and therefore it is an energy saving process. Otherwise, the separate pathway which starts from the stretch or which starts from the individual molecules is called de novo pathway. So, there is a de novo pathway, and then there is a salvage pathway. So, this is very critically a salvage pathway specific organelle in the cytoplasm of a cell. So, proteins are of course, we are talking about a non-self protein is taken into the cell, and this is just an antigen presenting cell which can express MHC1. So, that is what we are trying to focus on. So, these antigens are taken by the antigen presenting cell, and again re-emphasizing, antigen presenting cell could be a dendritic cell, could be a macrophages, or could be a B cell, and all the nucleated cell will express MHC1. So, once the antigens are taken by the cell, these are degraded by proteasome, and we can see that these are the smaller fragment of peptides which are coming from these antigens. So, these are all specific epitopic regions which are coming out, just to showcase. These are further has to go to some place, right, and these peptides are transferred to endoplasmic reticulum with the help of this TAP protein. We will see that structure of TAP and what it stands for a little later. Just to grossly look into what is happening, it is a kind of a channel protein. It is an active protein. It requires an ATP molecule in order to transfer this peptide into the ER lumen. In the ER lumen, we already have this MHC1 molecule which is preformed. Again, we will see how these are formed, and these peptides are loaded over this preformed MHC1 molecule in this alpha 1 and alpha 2 fold. So, this peptide can be loaded here in between this alpha 1 and alpha 2 fold, and then it is dragged towards the surface so that it can display the peptide properly. So, one can see that these peptides which are presented in the MHC1 molecule is budding out through this smaller vesicle like structure. These are going and fusing with the cell membrane, and the MHC loaded peptide can be displayed outside. Once these are displayed, the CD8 specific T cell which we have discussed in the past, that MHC1 pathway will very specifically attract CD8 specific T cell or cytotoxic T cell. Now, these T cells are specific cells of T lymphocyte which can express CD8 receptor on the surface. So, these are simply a protein which are expressed on the surface of T cell. So, these proteins which are expressed can give a specificity of a cell that these are cytotoxic T cell, and this can identify this molecule loaded on MHC1 and then start a cascade. So, there will be a downstream signaling pathway which can activate this cell type to do further processing, so that this antigen which entered inside the body can be recognized as non-self to be degraded by the immune system. So, ideally this is a very synchronized pathway, and one can see that this pathway is going through multiple ways in order to present it to the CD8 specific T cell. So, what CD8 specific T cell is identifying is the signature molecule on the peptide, and who is displaying it is MHC1. So, the two things which we need to understand here, that the proteins are always endogenous. So, these are always coming inside the cytoplasm, and the second thing is CD8 specific T cells are activated through the MHC1 pathway. So, just summarizing the whole pathway, we are trying to understand the endogenous pathway. This is also called as the MHC1 class pathway or class 1 MHC pathway. The antigens are taken into the cytoplasm, or the antigens are processed in the cytoplasm by a virus infection, and always these antigen processing are present in the cytoplasm to be understood as an endogenous. So, these are not exogenous antigen. So, these antigen uptake can degrade this bigger antigen into smaller fragment by this structure called proteasome. This smaller peptide can transfer to the endoplasmic reticulum with the help of this transporter protein called TAP, and this can load the protein on the MHC1 preformed MHC1 molecule which are there in the ER. So, one can always remember that the production of MHC is happening in the ER level, and then these are transferred to the surface through this vesicle based transport system, and then displaying it out to the T cell receptor, particularly the CD8 positive T cell receptor. So, this is one complete biology of endogenous pathway. Let us try to understand few details which are important here in order to understand this endogenous pathway. So, one thing which we have discussed, that the receptor. So, the receptor on the CD8, so suppose these are endogenous molecules which are presented through this MHC1 molecule. So, let us try to understand the T cell receptors. So, the T cell receptors are specific protein present on the T cell. So, we just now looked into that. Once the MHC loaded peptides are coming, they are identified by this T cell receptor which are present on the T cell, and these are positive for CD8 expression, and these can recognize this molecule critically. So, let us try to see that how this T cell receptor looks like. So, we could look into the structure as there are two chains, same alpha and beta, and again these are divided into variable and the constant region separately. There is same recombination of the fragment in the level of gene, that is leading to variation in the T cell receptor to bind various types of antigens. So, this is simply an antigen binding site which are coming from the MHC1 loading structures. Now, this constant and variable region is similar like immunoglobulin family which we have discussed, and therefore, we often see in the textbook that these are called immunoglobulin receptor family. So, these are all look like immunoglobulin molecule, and therefore, these are all Ig receptor families. Again, these are transmembrane region and then downstream cytoplasmic tail which could lead to signaling cascade to activate the T cell once there is a binding, otherwise there is no activation. So, this is T cell receptor which we need to understand in order to figure it out, that how this T cell specific biology is taking place post binding to the antigen. Next is the structure which we have discussed called TAP, and TAP stands for transporter associated with antigen processing. Remember, we discussed that the TAP is giving entry of peptide to the ER level, right, or ER inside the ER lumen. So, these are the proteins which are present on the ER membrane. So, what is happening to this TAP? So, these are transporter, and again these are heterodimeric protein. And what do we mean by heterodimeric protein is there are two distinct type of proteins, and these are two different varieties of amino acid chains which are present. One is called TAP1, the other is called TAP2, and therefore, when there is an assembly of TAP1 and TAP2, we can call it heterodimer. Otherwise, we simply call it homodimer if there is assembly of TAP1 to TAP1 or TAP2 to TAP2. But since there is assembly of TAP1 to TAP2, we call it heterodimer. these are located on the membrane of ER. So, let us say this is ER membrane, and this is giving passage of peptide to the lumen of ER in order to load it with the MHC1 molecule, which we will see a little later. So, let us try to understand this structure, because again there are a lot of things which are associated with TAP, and we need to understand this critically. So, these are transmembrane, there are domains which are multiple transmembrane domains are there. So, this is an N-terminal domain which we can see here, and then we could also look into that the whole structures are actually giving multiple ways of the protein. So, the protein foldings are there, and then the bottom is the ATP binding site. That means it is an active process. Once there is a binding of adenosine triphosphate, it is an energy driven process. The peptide can enter inside the system. So, let us try to understand this TAP protein called transporter associated with antigen processing. As we say that these are heterodimeric protein. So, there are two complexes which make this protein, one is called TAP1 and the other is called TAP2, and we could see that these are cytoplasmic area which is there in the cell, and of course, these are lumen of the ER. So, the peptides which are coming from this side can go to the ER lumen, where they can bind to the MHC1 molecule. So, the protein is very complex, and we can look into that is the individual protein is having an N-terminal domain, and multiple round of helical structures are there in the protein, and finally, the cytoplasmic domain or the C-terminal, the carboxy terminal domain is having a ATP binding site, which will allow the entry of the antigens to the lumen of ER with the help of an energy driven process. So, the ATP, adenosine triphosphate, convert into adenosine diphosphate and provide a phosphate group in order to give an access of antigen to the ER lumen. So, this is an energy driven process. We need to also understand that there are membrane biology, and these specific peptides which are generated through the proteasomal degradation is basically loaded by these structures. Now, since these structures are very important, and any deviation in this structure could lead to severe reduction in the MHC1 surface expression. So, basically these are important proteins which are present on the surface of ER, and if there is a deviation, we could see that these antigens are not going properly to the ER lumen, and therefore, these are not processed through MHC1 pathway, and we could understand that there could be a reduction in the MHC1 expression if there is a mutation in the TAP genes in the human body. So, these can lead to multiple chronic infection, and occasionally the autoimmune diseases. For example, if there is no MHC1 expression in a proper way, we could see that the antigens are not recognized properly, and the second, if there is a faulty expression of TAP1 and TAP2, there could be a possibility that the mispresentation of antigen can happen that could lead to autoimmune response, because MHC1 can raise the peptide specific autoimmune response when the self peptide can be processed through this pathway. So, this is another important protein which is associated with the MHC1 pathway. Let us move on and see that what are else in the MHC1 pathway. Now, what we understood is TAP is important, and there are three functions of TAP. One is peptide transport, which we understood that it can give access to the peptide to the ER lumen. The second is the MHC assembly, and that is what we said, that anything deviation in the TAP1 and TAP2 could lead to misassembly of the MHC1 molecule. Now, why this is important? There are three more proteins which are present on the ER. One is called tapacine, the calreticulin, and ERP57, which is endoplasmic reticulum protein 57. These are chaperones. What are molecular chaperones? Molecular chaperones are specific proteins which are present in the cytoplasm, and these are helping the cellular proteins to fold properly. So, these are assistant which are present on the cytoplasm. They assist the protein folding in a proper way. So without chaperone, you don't see a functionality of a protein in a proper way. So chaperones are a molecular folder, so they can fold the protein in a proper way. So these are the three molecular chaperones which are present on the ER. One is called tapacine, the calreticulin, and the P57. We will see its function little later, because these are responsible for MHC assembly, because we do remember that there are two distinct chain of MHC, one molecule, one is alpha and the other is beta 2 microglobulin. So these two chains are present on ER differently, and these chaperones can help them to assemble it in a proper way, and therefore we can call it the folding of the protein is important, and this folding can be done by these chaperones. And finally, the immune surveillance, because it can detect the viral specific infection, the tumor specific protein presentation. So these TAP proteins are very important when it comes to MHC1 specific biology of the pathway. So just remember that these are involved in protein trafficking or protein transport. The second is it can assemble the MHC molecule on the ER surface with the help of multiple chaperones, and few of them are listed here. And the third is they can help in immune surveillance, because they can detect virus infection and also tumor specific antigens which are non-self in nature. Okay. So let's see again this pathway in little more depth. So what we have seen that the endogenous antigens are produced, and these are loaded on MHC1 and presented to CD8 molecule. But what is happening internally? We have looked into the TAP structure. Let's see little deeper about how things are happening in the ER. So ER structure we need to focus, but let's see what are the two steps which are beyond ER. So one is the cytosolic part, where the protein is getting degraded into endogenous protein. These are getting into proteasome, and then they are fragmented into peptides, and these peptides are again loaded on the structures which are called as MHC1. Now what is happening in MHC1 synthesis or chain synthesis in ER? Now let's see the step number 3. So once the MHC1 alpha chain is synthesized, so here you can see that alpha chain is there, it is there in the ER membrane, and these beta 2 microglobulins are coming into picture, because unless until the beta 2 microglobulin come along with the MHC alpha chain, you don't see a complete MHC1 molecule. So these are coming close to the MHC1 with the help of all this chaperone which we discussed, the calreticulin, ERP70, ERP57, and also there is another one, calreticulin, calnaxine. So apart from that, so tapacine is little below the step, and therefore we will just see that these three chaperones, ERP57 and calreticulin, and one additional name is calnaxine. These two are associated protein actually in the cytoplasm. Calnaxine and calreticulin is actually a protein which can modulate based on the calcium stuff in the ER, and ER is a site of calcium channeling. So that is beyond the discussion of this lecture, but just for your knowledge, these are calcium modulated proteins. There is another protein which are modulated to calcium called calmodulin, which is calmodulin, which are present in the cytoplasm, but here we have ERP57, calreticulin and calnaxine. Now these three proteins can assist, so these can assist in proper folding of beta 2 microglobulin towards the MHC1 molecule. So MHC1 molecule is getting proper shape with the help of these molecular chaperones, and once the MHC1 molecule is taking proper shape, and then TAP protein is coming into picture, the transporter of antigenic peptides, then this tapacine is actually another assistant which is present here that helps to load the peptide from TAP to the MHC1 molecule. So this is a kind of an assistant that load the peptide, the passed peptide from the TAP1, and load the peptide on the MHC1 molecule. So the whole picture is the ER is critically important in MHC1 expression. There are two major players here. One is called TAP protein which is giving an axis of antigen, and the second is MHC1 molecule. Now, how these are assembled? MHC1 molecules are assembled with the help of multiple chaperones to form a complete working model of MHC1, and the second is TAP is functionally viable in the presence of tapacine. So, once the tapacine is there, the peptide entered through the TAP protein can be loaded properly on the MHC1 molecule, and the functioning of each and every component of ER is very important, and without that we do not see a processing of antigen in a proper way to MHC1 molecule. So, once these are there, they are going to the Golgi apparatus, the things are budding out and then represented to the cell surface in a proper way, and this can be identified by a cytotoxic T cell population from outside. To just give a signaling cascade, and we also looked into the TCR, how this alpha and beta chain of TCR is involved in recognizing this antigen. So, this is complete picture of endogenous expression of the peptide and its presentation by MHC1 molecules. So, just to summarize the whole picture, we will look into this detail of downstream cascade of how these cytotoxic T cell are activated in our later classes, but till now what we have understood that there is an activation of CD8 specific T cell by MHC1 class. So, these are antigen presenting cells which are processing this antigen through MHC1, and TCR is coming which are positive in CD8 molecules. So, this is a receptor for T cell, and this TCR once identifying this MHC1 class is getting activated. Once these are activating, these can clear this antigen from the body tissue or body surface, or this can clear the antigen from the body system effectively. So, this is just an unique way the viruses are cleared from the human body. Most of the time the endogenous antigens are coming from viruses, and the nature of those antigens are also specifically endogenous in nature. So, we will try to look into the exogenous pathway, which is MHC2 class, and we will entirely do a different thing, all set of structures in a different way. We have looked into the MHC2 molecules separately. So, we will try to understand this exogenous pathway in our next class. Thank you.